So I'm a neurologist, but I do genetics as research, and what I wanted to try and get into today, yeah, I think so, that's it. Yeah, that's it. Is what genetics has to offer, how we think about, thank you very much, the future of neurology and the future of designing therapies. And I am, of course, a bit biased in this, but I think it's fundamental to how we understand the disease. We're going to have to, we all have genetic variation. All of us sitting in this room now are slightly different to the person next to us. Not massively different, but slightly different. We are all harboring disease-causing mutations. We've all got pathological mutations in our genomes. It is a strong belief by, I think, most people in the medical fraternity that actually some of those variations, some of that variation in the genome is predisposing our risk to disease, and as Dominic alluded to it towards the end of his talk there, actually influencing how we do or don't respond to drugs, and how we dissect that is a real challenge. So there are two broad ways we think about genetics and genetic discovery. You can map it, and I'm not going to talk about the ways we do that, but we can either use linkage, which is a way of getting the high penetrant, the family that Tim was talking about earlier, about an hour ago. That's something that goes through the family, and everybody who gets that mutation gets that disease, so it's to be highly predictive. And then you have this disease risk. Us in this room, some of us will be predisposed to diabetes, some towards hypertension, some towards cancer, and that is one of the modest, commonly occurring genetic risk factors, but they up our level substantially, and we use a different sort of study called a GWAS for that. But a major technology that's come on in the last 10, 15 years is being able to sequence the genome. When I was, 15 years ago, to sequence even a small bit of DNA cost a huge amount of money. Now it's relatively trivial, and I'll talk about that halfway through. The way to think about it is graphically displayed here. So at this end, these things are very rare in the community, one in tens of thousands quite often. But when you have this change in your genome, you've got a very high prediction of getting a disease, Huntington's disease, some of the dementias Tim was talking about. At this end, you've got the rest of us. These occur in most of us at some level, and these increase our risk for the commonly occurring diseases. And there probably isn't a commonly occurring disease in which genetic factors aren't having some role. Parkinson's, Alzheimer's, ALS, epilepsy. Even our recovery from head injury has genetic determinants to it. I'm going to illustrate my talk with Parkinson's disease because that's what I work on, and I think it's proven to be very useful. So Parkinson's disease is common. It affects lots of people, famous and not so famous people. But the reason I think the genetics has worked for this disease, because it's not a very heritable disease. There's those in the room who've got Parkinson's disease, if there is anybody, the chance of your relative having it isn't that high. It is not a very genetic disease, but it's turned out to be useful because the phenotype is very solid. If you have the core cardinal features, you've got a very high predictive chance of having that type of pathology in this bit of your brain. And because that's reasonably clean, the genetic signatures that there have been able to be pulled out by the techniques that we've had available in the last 10, 15 years. And that's why it's been a success. So if you think of that graph I showed you, you've got the high penetrant mutations here, you've got the commonly occurring ones down here. You can see Parkinson's disease has really filled in nicely in the last 10 years or so. So when I started doing this work 20-odd years ago, people laughed at me in the genetic lab because I'd picked a non-genetic disease. I wanted to work on mitochondrial disease or ataxia or something that clearly had a gene in it, and yet it's turned out to be reasonably fruitful. There are more to come. This is published in 2014. We're about to publish the next iteration of this, and there's a few more genes to come out. I briefly want to go through three issues in the talk today with this background of these genes. Can we use what we've found so far to get a gene risk score? What else is there to be discovered? And importantly, the main bit of the latter part is how we use this, how we might think about this to use for novel drug validation discovery. And that's a new method I want to go through in a little bit of detail. So gene risk score, what we're trying to say here is if you've got, let's say there's a risk with all of these are just names of different genes. If you carry the risk allele, the risk variation in every one of those, whatever the number is, 20 loci, 20-odd loci, what does that do to your risk? How much does that amplify your risk? And also, if it does, can we identify those people and use those people in our studies? Because they're exactly the group we're going to need to be doing our pre-symptomatic trials in, because they're the ones at greatest risk. That's what we're trying to get at. So that's those genetic factors spun out here on a wheel of the various types. And you can do something where you can plot out an area under a curve of this analysis here. If it's a diagonal like that, it's random. You're doing no better than just guessing whether you're at risk or not at risk. Anything to the left of the diagonal is better than chance. And once you get over 90% or 0.9, you're getting to be quite useful. So if you put all those gene risk scores together, gene factors together, and we take into account a few other factors, including sense of smell, family history of Parkinson's disease, and a couple other things, we get a score of just over 90%, which sounds good. It's a start, but it's still not very good for predicting people. We cannot use this in the clinic. We cannot yet roll this out and use it in any sensible way, because even if you carry all those risk variants in your genome, you've still got a greater than 95% chance of not getting Parkinson's disease. So it's not very good yet at giving data into the community. But we want to strive to get that better in the future. I think I'll pass over that for now. If we then think about what's left to find, you can do something called a heritability analysis, where you measure basically how heritable a disease trait is. And we did this a few years ago now, and you can see that the heritability estimate was around 28%. So, i.e., the risk of getting Parkinson's disease is about 28% encoded within your genome. That's roughly how you can think about it. When we did the analysis in 2012, we'd been able to explain about 5 to 8% of that. So there's a long way to go to explain the rest. This concept of missing heritability has bedeviled the field for about 10 years now. Why is it, with all these thorough techniques, the GWASs, the linkage, the this, the that, the sequencing, are we not getting much better capture? And I want to give you a couple of illustrations that we in the consortium have made in the last couple of years. So some of it could be just even more number of genetic variants still to be found, and that's certainly true. Some of it is that the GWAS studies are not good at picking up certain types of mutations, and these are called copy number variations. Us sitting here in this room now have abnormalities in our genome. We have duplicated some genes, we've deleted other genes, and we're sitting here perfectly healthy or reasonably healthy, and it's not seemingly having an effect on us. But sometimes it will have an effect on our disease, and we need to be able to capture that. There are other things I haven't got time to go into, but I'm happy to answer questions about. So this is a bit of history. So this gene for this condition is called dopa-responsive dystonia. It's a very rare condition, autosomal dominant, 50-50 risk to at-risk carriers to carry transmission of first-degree relatives. If you get this disease, you have an abnormality in your synthesis of dopamine in your body. It's a great disease to diagnose a neurologist, because if you do diagnose it, you can give people L-DOPA, Sinemet, the treatment for Parkinson's disease, and they respond perfectly well for the rest of their life, because it's not a degenerative condition, it's a biochemical defect. And so you bypass the biochemical defect, and you're on their Christmas card for life. It's a very satisfying diagnosis to make. But sometimes people in those families look a bit Parkinsonian. And so the question arose, was that a Parkinsonian phenotype of DRD, or are they actually getting Parkinson's disease? And there were a few random reports out there, and nobody was quite sure. Well, a couple of years ago, we found 4 families. These guys lower down the generations have classical dopa-responsive dystonia, and these senior people have Parkinson's disease. But they don't just have DRD-like Parkinsonism, they have what you might call standard Parkinson's disease. They look like Parkinson's disease patients. They continue to degenerate. They get the movement dyskinesia you see in Parkinson's disease. They have some of the cognitive issues you see in Parkinson's disease, and they have abnormal dopamine uptake, which is normal in DRD and not abnormal. So they looked all the world like Parkinson's disease. So you see that once in a family in a rare disease, it just happens. Parkinson's disease is common enough. See it 4 times, it seemed a little bit too unlikely. So we were able to do 2 things. We'd sequenced a lot of what we call exomes. These are the coding bits of the DNA. So we could go back through this database. So this database has been done already, but we could focus in and say, what is the hit rate of rare variation in the DRD gene, in those things? And we are predicting that it's going to be in excess to what you'd expect given the size of the gene. And that's exactly what we found. And also, we could do a GWAS. And you can see that in the advanced GWAS, the mega meta-analysis, the GCH1, the gene, lights up. So both in rare variation exome sequencing and common variation, we get a double hit. That cannot be by chance, and it seems that something in this biochemical pathway predisposes some people to not just getting DRD, but actually having degenerative disease of the nigrostriatal pathways and getting Parkinson's disease. To illustrate the copy number variation, this is the commonest small deletion we have. It's on chromosome 22, the long arm, that's Q, at 1.2, and it's relatively common. And when you get it, you get a condition called DeGeorge syndrome. It's quite in its fullest sense, it's quite a full-on disease. It can produce quite a lot of difficulty with cardiac disease, with calcium metabolism, and it's also been associated with schizophrenia. And there's been 1 or 2 case reports, again, of perhaps being something to do with Parkinson's disease. Well, again, a bit like the story I had for DRD just a moment ago, we did the same retrospective analysis targeted on this region, looking for evidence of copy number variation of 22q, and that's what we found. We validated it with a second method to show that it wasn't just a quirk of the method we were using for ascertain the data, and you can see the p-value down here, probably, which is 0.00 something. It's very low. So this is rare, but again, a cause of Parkinson's disease. Now, the number of genes deleted in this 22q region is really quite large, and so the work is now on to work out what it is about missing that bit of genome that predisposes you to get Parkinson's disease, and does it have anything to say about schizophrenia? So it's challenging. So the third thing, and the thing I want to really focus on, is what genetics can do next. We undoubtedly will have larger scale whole exome, the coding bit of the DNA, about 1%, 1.5% of our genome, and whole genome studies. And I want to talk about a technique called Mendelian randomization to get onto drugs. The reason we're going to get larger studies is really this: it's technological advance. So, when Dominic first came across to UCL, it was around then, wasn't it? It cost $100 million to sequence a genome. The first genome took 10 years, probably cost $3 billion, $1 a genome, $1 a base pair, sorry. So there's 3 billion base pairs in each of our genomes, and took multiple labs 10 years to do. The costs came down with Moore's law. Moore's law, Moore predicted in the 60s that you'd have double the number of transistors on a bit of plastic every 18 months, and now we have chips. That's why our smartphones are cheaper and more powerful. And the cost of sequencing mirrored that all the way down till about 10 years ago. And then the cost fell off a cliff because of a technological development. And 3 years ago, it reached $1,000 a genome. And in fact, I just got an email this morning. They're offering it in the UK now for £600, which just with the devalued pound is probably still $1,000, but that's Brexit for you. But we won't get into Brexit just now. So it's really cheap. It's as cheap as an MRI scan to sequence a genome. It is really that cheap. So capturing genetic variation is trivial, and it's going to become increasingly trivial. We are not going to be doing blood spots on kids like we do at the moment for some rare diseases. We will be sequencing genomes. I don't know when that will be, but it will be within the next few years. That will be the norm. People will walk around with their genomes available to their healthcare researchers. Now, the problem illustrated here is that actually computing power, despite it being great and improving, hasn't kept up in price. I mean, this is not a later scale, but the principles are the same. It is now cheaper to sequence a genome than it is to store it. So logically, you should sequence it and throw it away and resequence it when you want it again, which obviously is crazy. We don't do that. But it actually costs more for us to pay in tape storage and computer storage than it does to sequence the damn thing in the first place. So we need bioinformaticians, we need that computing insights and quantum computing and so forth to really make sense of this data, and we need good clinical observation. All right, I want to talk about the last point I want to make now, and this is a technique. It's been around about a decade now, called Mendelian randomization. And I first want to illustrate it with a study that we've just done, and it's due to be published in a couple of weeks' time. And I won't go into this in great depth, but essentially what you're doing is you're getting genetic variation captured here and being able to predict an outcome, in this case, Parkinson's disease. So the thing that we started with was BMI, body mass index. Clinicians know that you rarely see a patient with Parkinson's disease who is anything other than on the slim side. You do constantly see, but they're slimmer than the rest of us. Now, is that because they've got a chronic disease? Is it because of the movements? Or is it something fundamental to the cause of the disease? And the literature is just not clear on that. This genetic technique is a way of getting into that causality relationship. Because what you can do, you can take a genome-wide association study, a GWAS, of tens of thousands of people of all body sizes. You can do genetic capture, and you can find out the genetic information that predicts their body weight. It doesn't predict it 100%. It's not all, we know weight is not entirely genetic. I'm not saying that. But we can capture pretty accurately and very robustly the amount of variation that predicts your weight. So then we can take those variables and then just ask that question of the disease control grouping and say, okay, we're not going to weigh the people. We don't care what their weight is. We're just saying what their genetic capture is. And we do that for Parkinson's disease, genetic variation, through the BMI as the cause of Parkinson's disease. And we see that actually, using quite large numbers here, 13,500 cases, almost 100,000 controls, these very large studies, that having an increased BMI of 5 kgs per meter squared, quite a big BMI increase, you decrease the risk of PD by about 18%. Now, it is one study. I'm not suggesting that people go out and tell people with Parkinson's disease to go and eat 2 more pies. It isn't, we're not that certain yet. But nevertheless, it shows the power of being able to ask complicated genetic questions in a causally related way. Now, I want to talk about drug discovery. And, stroke has improved massively in terms of therapeutic approaches in the last few years. Somebody had a question about MS a little while ago and so forth. Neurology has entered the therapeutic arena, but neurodegeneration has proven very difficult. There are no disease-modifying drugs for Parkinson's, ALS, or Alzheimer's disease, and it's hugely important to us as developed economies. We spend more money on dementia than we do on cancer, cardiovascular disease, and diabetes combined, but 98.5% of it is on care because it's so disabling and so chronic. So we have to do something about it because we can't afford it as an economy. So it's a major driver. But if you think about the pathway to developing drugs, you have your disease hypothesis. So people like me come up with an idea about what causes a disease. We target select based on our either genetics or mouse models or fly models or whatever it is we study. And then we go through a whole bunch of studies right through to development. When you're down here, it's incredibly expensive. You're not talking millions of dollars. At phase 3, you're often talking tens of millions or hundreds of millions of dollars. And by the time that all the drugs fall out, it's often well over $1 billion to get a drug to market. But most of them fail. And they're failing now at this stage. Now, the drug companies and researchers in this area over the last 20, 30 years have addressed some of the problems used to be the study design. But people know how to do that pretty well now. That is not the major problem. The major problem is target selection. We're just very bad at getting the right target. And we're very bad. If you start there, the whole thing is not knackered. I was trying to think of a polite word to say. And very expensive. And so can we do better? That's the challenge. So the standard drug therapy, of course, is the randomized controlled trial. And what you do there is you get your patients, a computer randomizes them, you put some into a placebo group and some into whatever intervention it is, talking about platelets before, and you know the intervention binds a target, and then you measure the outcome. And it's very good because it's random and you correct for all sorts of confounders and biases and so forth. Our genetics are nature's randomized control trial. Our alleles, our variation, which are mothers and fathers had were randomly given to us at conception. We don't know what they are, of course, but we now have ways of capturing it, as I keep mentioning. So we can capture that. And what you have to remember here is that drugs bind to a target, and that target almost always has a gene which encodes it. Makes more of it, makes less of it, makes it more available to be binded to, and so forth. And so if you understand the variation that underpins that target, you can find variation that reduces the activity of that protein or increases the activity of the protein, i.e., it mimics the drug. It may not be as potent a manipulator of that target, but it's doing it anyway, and those alleles that do that were randomly assorted at birth. So I'll illustrate that with this model here. You've got a compound, it binds to a target. That target is encoded by a gene in our genomes, and you get a target effect in the outcome. And of course, the classical one would be a statin. You've got the enzyme, you've got the statin binding to it, on-target effect, intended outcome of lowering cholesterol and better cardiovascular and cerebrovascular outcomes. Well-established pathway. This is the randomized controlled trial bit on this side, and that's what an MR trial would look like. So you've got the randomization, you've got the target molecule here, you're binding to it, and you're getting the effects. You're lowering LDL cholesterol by 1 millimole, you're reducing your risk by 25%. We've got variation in that target which does the same thing. It doesn't do it by as much, so it lowers LDL cholesterol by about 1/10, as much as that, and it reduces your risk by about a quarter, so 6% rather than 25%. But the advantage of this is, this would have cost millions to do, millions and millions and millions to do. This has been done for us. There's already GWAS data out there. So if we had not known about statins through the other biology that had been discovered over the last 25 years, we can do this in a computer. We can do this from taking GWAS data, we can understand the variation in the target of this molecule, and we can ask the same question, and we would have found the same effect, and we'd have seen that this was going to be a good targeting. So, of course, the obvious challenge is, can we use it to discover new drugs for neurodegeneration and neurology? I think one of the really exciting opportunities is what we call repurposing. This, again, is illustrated by cardiovascular disease again. Rheumatoid arthritis, immune disease. There's a monoclonal antibody which binds to this receptor here called IL-6 and improves your outlook for rheumatoid arthritis. And it's been approved. It's been through all this stuff. It's had all that expensive stuff paid for. So it's a molecule that's known how to be safe, or biologic that's known how to be safe. So can we repurpose it and introduce it here without having to do all this bit? So you want to know, what's the chances of this working for cardiovascular disease? Well, there's a literature now that's showing that inflammation and immunity is important in cardiovascular disease. There is actually a literature showing that inflammation and immunity is important for neurological disease. So you could equally have here, but I haven't got the data to show it yet, that we could have Alzheimer's disease, this little bottom panel, or Parkinson's disease, or ALS. It would be equally logical to have a go at this genetically. And in fact, that's what my group are doing at the moment. So what you have here, again, is the drug intervention bit on this side of the slide, interfering with IL-6, with the antibody or the placebo, and you get a better outlook, lower disease activity, or same disease activity. And you get a biomarker. There's a phenotype, a signature of biomarker with increased IL-6, decreased CRP, and so forth. Here, we know the variation that we can have in IL-6R, this polymorphism here. You don't have to remember the number, it doesn't matter, but that is doing exactly the same thing to a lower impact. It's not as powerful as the drug, but it's doing the same thing, and you can see the signature is identical, and the outcome is the same. So this is a strong argument that this monoclonal antibody should be applied at phase 2 into cardiovascular patients, and that trial is now going ahead. And I think we need to be doing these sorts of studies for all our commonly occurring neurological diseases. So final slide. Where do I think we're going? I think genetics clearly has something to offer. I think it's a way of generating molecule and molecular pathways. That's what a lot of what my lab and group do. So we find genes for things and we try and work out how they fit together to cause Parkinson's or other diseases we're interested in. But interestingly, we can start to use the human as the animal model. That's really the essence of my talk, is that the translation from mouse to human has been poor, in Alzheimer's and Parkinson's. It's not really worked very well. And I think wherever possible, we should be looking at doing the studies designed in humans. And to do that, we need to find at-risk individuals. And it seems logical to me the first place to start are defining those who are genetically at risk and then putting them through what we call experimental neurology. Thanks very much.